Hello all, welcome to F.Y.B.Sc class. Today we are going to discuss about the types of bacteriological media from the unit Microbial Cultivation and Growth of F.Y.B.Sc course MB122 Microbial Cultivation and Growth. The learning objectives of this lecture. At the end of this lecture, students are able to understand the different types of the media which was used for the cultivation or the isolation of microorganisms. They will understand the uses of different nutrient media. Now, the cultural media which are used in microbiology, they are classified into several categories and the categorization will based on its physical state. Liquid media, the first state of the bacteriological media. So, those media who have liquid consistency, they can be called as liquid media. Second type of the media which are frequently used in microbiology lab are the solid media. As their name indicate, we have to add solidifying agent like agarose, agar-agar powder, gelatin. Because of its gel-like nature, they will confer solid consistency to such a kind of the media. Mainly, agar-agar powder is most commonly used for as a solidifying agent. Next type is the semi-solid media. Now, if the solidifying agent, if they are added in low concentration, then the media will confer semi-solid consistency. The use of the liquid media is to obtain the pure cultures, while the solid media can be used for isolation of pure culture and to count or estimate the bacterial population from different samples. In addition to that, the second classification of culture media is based on their composition or the ingredient which they have and their uses. So they may be defined media, complex media. Third type of culture media is the selective media, differential media, enriched media, enrichment media, synthetic media, indicator media, anaerobic medium, assay medium, maintenance media, transport media, minimal medium, complete medium. All are the different type of the media which are used in microbiology lab. So let's discuss about the defined medium, which was also known as the chemically defined medium. Chemically defined medium are also known as synthetic media. Now, for the preparation of any synthetic media or chemically defined media, we have to make the use of pure chemicals. So, as we are making the use of pure chemical, we are aware with the exact chemical composition of each and every chemical which we are using for the formulation of synthetic media. Now, the content of these media, they are highly reproducible. They are chemically defined media are generally costly as because we are making the use of pure chemicals. Such a kind of the defined medium can be used for the cultivation of autotrophs. We can also make the use of chemically defined medium or the synthetic medium for the metabolic studies of heterotrophs. Next type of the media are complex media. Complex media, in that case, we are making the use of raw material for the formulation of such a kind of the media. And therefore, as the raw material consists of crude ingredients, we are not aware with the exact composition of the chemicals which are present within that media. So, in case of the complex media, exact composition is not known. They are not reproducible as we are unaware with the exact composition of the chemicals. But as compared to the defined media, complex media are cheaper, easy to prepare, and they include ingredients like the peptone, meat extract, yeast extract, agar powder. All are the ingredients of the complex media. Such a kind of the complex media can be used for the cultivation of heterotrophs. Three commonly used complex media are: first one is nutrient broth, second one is the tryptic soya broth, and third type is the McConkey's agar. Now next type of the media which is used is the selective medium. Now, selective medium, it is a solid medium and it will consist of selective or inhibiting agent. Because of the presence of that selective or inhibitory agent, such a kind of the media, they will allow the growth of desired organism to be take place. In addition to that, because of this selective agent, they will not allow or they will inhibit the growth of unwanted microorganisms. Examples of selective media are the McConkey's agar. We can use them for the isolation of intestinal bacteria. Second example of the selective medium is the mannitol salt agar, which can be used for the cultivation of Staphylococcus aureus. Next type of the selective media is the cellulose agar, which can be used for cellulose degrading bacteria. Next example of selective media is the cetrimide agar that can be used for the cultivation of Pseudomonas aeruginosa. Now, selective agents like the McConkey's agar, they will consist of bile salt or dyes like the basic fuchsin or the cristobolate. Now, such a kind of the dyes or the bile salt, they will favor the growth of gram-negative bacteria. At the same time, the gram-positive bacteria, they will not tolerate bile salt or the dyes, and therefore such a kind of the media which consists of bile salt or the dyes like basic fuchsin and the cristobolate, they can inhibit the growth of gram-positive bacteria. Examples of such a kind of the agar, selective agar, are the endo agar, eosin methylene blue agar, McConkey's agar. These three types of the agar, they are most widely used for detection of E. coli and related bacteria which are present in the water supplies. Next one is the differential medium. Now, such a type of the medium can be used for differentiation between two groups of bacteria. Examples of such a kind of the differential medium are McConkey's agar. Now, by using this Microbial agar, we can differentiate between two groups of bacteria, lactose fermenting and lactose non-fermenting bacteria. Now, for the differentiation between these two types of the bacteria, the Microbial agar should consist of lactose and an indicator system, that is the neutral red. Next type of the differential medium is the blood agar. So, blood agar can be used to differentiate between alpha, beta and gamma hemolytic bacteria. Now, if we see the Microbial agar, it can act as a both differential and selective agar because the Microbial agar consists of lactose as a carbon source, and therefore those bacteria which have a tendency to utilize lactose, they can call as a lactose fermenting, while those bacteria who are unable to utilize lactose, they can be differentiated as lactose non-fermenting bacteria. In addition to that, for the detection of lactose fermentation or the utilization by particular group of bacteria, we are indicating or incorporating an indicator within Microbial agar. So, neutral red is one of the indicator which is incorporated into Microbial agar, which help us to detect the fermentation of lactose. So, if a particular organism it utilizes lactose, then acidic conditions will be developed or formed and the dye will turn pink in color, and therefore the colony will also uptake and appear pink in color in case of the lactose fermenting microorganism. While lactose not fermenting bacteria, they appear white in color. Next type of the media are the enriched media. Enriched media, they are solid media, but they will support the growth of fastidious bacteria. Now, such a kind of the enriched medium can be prepared by adding the substances like the blood, serum and the egg to the basal media so that the nutritional requirement of the bacteria can be reached and the growth of the bacteria will take place. Examples of enriched media are the blood agar, chocolate agar, loeffler's serum slope, Algae medium. These are some of the examples. Blood agar is an enriched medium as in case of the blood agar, whole blood supplements can be provided which can act as a basic nutrients. In case of the chocolate agar, in that we have added heat treated blood which turns brown in color because of the heating treatment, and therefore it will give the medium brown color, and therefore such a kind of the media will be called as the chocolate agar. Next type of the media are the enrichment medium. Enrichment medium, it is a liquid medium which consists of selective agent. Now, such a kind of the medium, they will allow the growth of specific organism to be take place. At the same time, they do not allow growth of unwanted organism. Such a kind of the media can be used to increase the number or proportion of interested or the desired bacteria in the mixed culture. Examples of such a kind of the enrichment media are the selenite F broth, terrathionate broth which can be used for the isolation of Salmonella. Next type of the media are the indicator media. Now, this media will consist of some chemical or indicator, and because of that indicator, we can confirm the growth of particular type of bacteria is take place or not. Example is the Wilson and Blair medium. Now this Wilson and Blair medium, it consists of ferrous sulfate. Species of the Salmonella, they have a tendency to reduce sulfate to the sulfide, and therefore they can produce black shine on the colonies. So black color colonies will indicate the growth of Salmonella. Next type is the anaerobic medium. Anaerobic, that is, it can be used for cultivation of anaerobic bacteria. Anaerobic bacteria are those bacteria which will not grow in presence of oxygen. Now, to promote or to cultivate the growth of anaerobic bacteria, the medium which we are designing, it should consist of reducing agents. Examples of anaerobic medium are the Robertson's Cook medium. Second one is the fluid thioglycolate medium. By using these two medium, we can cultivate anaerobic or those bacteria which do not require oxygen for their growth. Next type of the media are assay medium. Now, to perform microbial assay of antibiotics and vitamins, we require some medium which can be called as the assay medium. So, for every microbiological assay, we require standard organism, and for the assay process, specific nutrient medium is suggested in Indian Pharmacopoeia. Now we have to select for assay medium such a kind of the medium that will support the maximum growth of standard organisms. Next type of the media are the maintenance media. Now, to maintain the viability and physiological characteristic of culture can be maintained by using such a kind of the maintenance media. Now, maintenance media, they should not supposed to support the rapid growth of bacteria, because we want to maintain that culture for the long period of time. If rapid growth of the organism is take place, then it may lead to the rapid death of bacteria. So while preparing the maintenance media, we should avoid the use of glucose. Next type of the media are the transport medium. Transport medium can be used for the temporary storage of patient sample from area of collection to the laboratory. So, the main use of transport medium is to prevent the death of organisms during transport. And in addition to that, if we are providing nutrient, then we can maintain the culture in viable or living state for long period of time. This media, they contain mainly buffer and the salts. Examples of such a kind of the media are the Stewart medium, which is a soft agar gel which consists of reducing agent that will prevent the oxidation, and charcoal to neutralize. Such a kind of the media can be used for the Neisseria gonorrhoeae. Next one is the buffered glycerol media can be used for the transport of Vibrio cholerae. Next one is the minimal medium. As their name indicate, minimal medium consists of minimum nutrients for the growth of microorganisms. As they are devoid or they will lack the growth factors like amino acids and the vitamins. Such a kind of the minimal medium can be used to grow wild type of organisms, auxotrophic mutants, those who are unable to synthesize their own growth factor are known as auxotrophic mutant. Minimal medium cannot be used for the growth of auxotrophic microorganisms. Last type of the media is the complete medium. Complete medium, as its name indicates, it will consist of all the nutrients as well as growth factors which are required for the growth of organism to be take place. As complete medium consists of growth factors, so such a kind of the medium can be used for the cultivation of auxotrophs also. These are the references. Thank you.